is made using bacteria. Who else can do it? There's an interesting story of, in general, most higher animals can't do this. Right? They don't have the pyruvate decarboxylase. Now there's one interesting exception to that. This is one of those things, I stumbled across this paper. I used to like doing offbeat seminars. You know, everybody else would do their graduate school seminars, class seminars on whatever their research was. I liked finding offbeat, only marginally connected things. Does that surprise anyone that I would go for the offbeat stories instead of whatever I was supposed to be talking about? And so one of the ones I stumbled across had to do with a certain genus of goldfish and European carp. Right? In the genus Carassius. You don't have to write that down, I just find it interesting. If it's a little goldfish, it's Carassius auratus. If it's a European carp, it's Carassius carassius. Right? So, goldfish, carp. But the deal is, these are the kind of goldfish or carp, especially these are northern European carp. Now what happens if you're a carp in a pond in northern Europe or southern Scandinavia? Or apparently Indiana in 2014.
It's cold.
It's cold. Cold enough that what happens to your pond?
Freezes.
It freezes. And when you get a layer of ice over the top of your pond, what happens to the oxygen exchange? Goes down would be the polite thing to say. None. It becomes non-existent. So under the ice, these ponds can go nearly anoxic for long periods of time. And so, how does this goldfish or carp live under the ice in this water that has almost no oxygen, but yet when the water thaws in the spring, they come out and they're a little sluggish, but still alive. And so people started studying this. So what they did was they put them in completely enclosed tanks, and they found that one of the things that the carp did was, in the fall, they ate a lot of carbohydrates and built up large supplies of glycogen, source of glucose. And so they injected them with radioactive glycogen, and then looked in the, what? And then they looked in the water for where the radioactivity came out, right? What kinds of molecules were being produced. And what they were producing was ethanol. They were turning the glucose into ethanol. So it's the only vertebrate group known to do that. That actually it appeared to be something going on in the mitochondria of their muscles. They don't have a pyruvate decarboxylase enzyme, but they were using, we'll talk about pyruvate dehydrogenase. Parts of the pyruvate dehydrogenase enzyme are closely related to pyruvate decarboxylase. And so apparently they're using, under these conditions of very high NADH, their pyruvate dehydrogenase enzyme can act like a pyruvate decarboxylase. They have an alcohol dehydrogenase that can reduce that, and it can produce enough ATP to keep their cellular processes going, to keep them sluggish but alive over the course of a winter under the frozen surface of a pond. So, if we had that capability, all you would have to do to have a party is, right? If you could hold your breath long enough, right? It would make, your muscles would make alcohol for you, and it would make weekends much cheaper. Right? So it's the only vertebrate group known to do
That's what I was thinking too.
What?
Melanie and I were like, oh, we could live underwater, or we could get drunk by ourselves. We were going a different route.
You can live underwater and get drunk in the cold. It'd be fun. Okay? So, it's the only group that can do that.
Little Mermaid.
That's what I thought he was going to say.
Me
and
too.
then-
Like
No.
Mark here and
No.
like we
Not that I know of.
I know you've said it.
I don't know.
Hmm?
But I know these
Oh,
European
no, not a bit.
So, yeah, I thought it was very cool. So that's mode one, making ethanol. So the whole point, whether you're a yeast, let's get to the other point, whether you are a yeast or a goldfish under a pond, why do you make ethanol as the product of this recycling of NAD? Well, let's go back, let's jump in our Wayback Machine. I'm so excited about the Peabody and Sherman movie because I grew up with Peabody and Sherman, so I'm excited.
I said I saw that because you've talked about that before.
Yeah. Oh yeah. So let's jump in our Wayback Machine and go back to last semester for some of us. We talked about transport processes. When we talked about ethanol in terms of membrane transport. What do we know about ethanol?
It can diffuse.
What?
It can diffuse.
It can diffuse across biological membranes without the need of energy or other help. So if you're a yeast who thought you were living high because you were surrounded by a sugary fluid, which then someone put in a container and closed that container off so you had no oxygen, right? What does the ethanol let you do? Right? What does this let you do? Keep doing glycolysis. But then where does the ethanol go?
Out of the cell.
Out of the cell. So the products don't build up inside the cell. You can let the products diffuse out of the cell into that large volume of surrounding fluid. Right? That's what it lets yeast do. Right? It lets them, with no energy at all, other than losing the energy they could get out of these two carbons, with no energy input at all, they can get rid of the products of this, which prolongs their ability to keep this going and keep glycolysis going. Same thing's true of these fish. Right? They're stuck under the ice. This is only providing them sort of bare minimal amount of ATP that they need to keep minimal cellular processes going. So this is made in those specialized muscle cells. So then what happens to it? It diffuses out where? Out of the muscle cells to where?
Bloodstream.
The bloodstream. But where does the blood go if you're a fish? Where? Goes to your gills. The gills are thin, have thin membranes, so you can get gas exchange. Through the gills, can we get ethanol exchange as well? Yeah. So they use the gills as a way to get rid of the products. So in each case, we're doing this as a way to generate the NAD we need to keep glycolysis going, while creating a product that the organism can allow to diffuse away without using the valuable available energy. Right? That make sense? What? It's pretty slick, yeah. It's almost like somebody thought this stuff out.
We have to diffuse through our lungs or something.
They could if our lungs were like,
It's not going to be all the way down there, diffusing all the way here.
No. I don't think so.
Go to here. Well, let's go to here. Talking a little bit about that decarboxylation reaction. The pyruvate decarboxylase and pyruvate dehydrogenase, we will find, uses this coenzyme, thiamine pyrophosphate, TPP. So when you're reading the side of your Pop-Tart box, and it says, you know, your vitamins and your B vitamins, and one of them is thiamine, that coenzyme is helping this step. It's used in the construction of this enzyme. So here we have thiamine pyrophosphate. We've got an amino pyrimidine ring, but we've got the thiazolium ring. And it's this thiazolium ring that's the important part for this reaction. And then there's a pyrophosphate group. In some enzymes, this pyrophosphate group is attached to an amino acid. In other enzymes, it's free. Okay? So we're going to talk about this. Yeah, let's go bigger. Bigger helps here. So, if you saw there, it said on this ring, this hydrogen ion is acidic. And we're going to get to pull back one of our great words. I was going to ask the word, but there it is, right? If we deprotonate this, then we get plus minus right next to each other. Whenever you have a Zwitter ion, where you've got a plus and negative on adjacent atoms, we call that an ylid, right? Remember we used the ylid in the Wittig reaction? We used a phosphorus carbon ylid in the Wittig reaction in organic chemistry. So here we've got a carbon-nitrogen ylid. And that carbanion that is stabilized by the positive charge here can act as a nucleophile on the carbonyl. Okay? So, ylid attacks as nucleophile, opens up that carbonyl, as happens when we get a nucleophilic attack, and we get this product. Now, let me see if I can bring this product up a little higher where we can see it. There we go. Now, here we go. This is where it gets cool. Because we've got now this C double bond N within the thiazolium ring. Right? And we said C double bond Ns are almost as good at stabling carbanions as C double bond O. So now suddenly this C double bond N makes this the alpha carbon and this the alpha beta bond that often becomes labile when we've got that there. So using this and having it react with the pyruvate makes this bond suddenly a bond we can break. And so this pair of electrons comes back here, creates carbon dioxide, right? This essentially comes back here and becomes a carbanion stabilized by this resonance. So these arrows they're showing you are moving us toward the more stable form with a neutral nitrogen and the unshared pair on the nitrogen. But if we resonate this back, we get this stabilized carbanion where there would be an unshared pair on that carbon. So we're using chemistry we've used before. We're getting a decarboxylation as a result of letting this double bond within the thiazolium ring act like our carbonyl in an aldol type reaction, one of our carbanion bond breaking reactions. Kind of sneaky, right? That was not slick, that was sneaky, right? We snuck in a C double bond N on them so that we could break that bond. Does that make sense?
What was that for?
This is the pyruvate decarboxylase. Right? This is the coenzyme interacting with the substrate in pyruvate decarboxylase. Okay? So now we've decarboxylated that. We protonate it with something.